Hello students, today's topic is immunodeficiency disease, especially HIV and AIDS. Introduction. Immune system is a body defense system which protects our body from the entry of antigen. And antigens are those foreign particles that enter our body. Those antigens that may cause disease in our body are called pathogens. Immune deficiency means failure of the immune system to protect the body adequately from infections due to absence or insufficiency of some components, process, or substances. There are certain reasons that lead to immunodeficiency. They may be either congenital or acquired. A congenital or primary disorder is one you were born with. Acquired or secondary disorder is those you get later in life. Acquired disorders are more common than congenital disorders. There are many immunodeficiency diseases. Among them, today we are going to discuss only AIDS. What is HIV? AIDS is caused by HIV. HIV is a retrovirus belonging to the Lentivirulidae family that attacks the immune system. Structurally, they consist of two identical strands of RNA associated with the matrix proteins, a double protein capsid, and a lipid envelope with inserted glycoproteins. The genomes include the usual retroviral genes, GAG, which codes for structural proteins, POL, which codes for the reverse transcriptase in its native form, or after fragmentation. The POL gene product has several enzymatic activities like polymerase, ribonuclease, endonuclease, integrase, and ligases. INF, which code for envelope glycoproteins. In addition, the HIV genome codes for a protease and several regulatory proteins. A gene located at the GAG-POL junction codes the proteases. The gene is read at different frame than the structural proteins, and the protease cut itself free from the large polypeptide chain generated from cell-expressed viral RNA and proceed to further split the remaining of the polyprotein precursor into several other proteins and peptides. The genes coding for regulatory protein includes TAT, also known as transactivator of transcription. Its product P16 or TAT protein is released by infected cells and taken up by both infected and non-infected cells. Finally, TAT protein has immunosuppressive effects on non-infected T cells. Number 2. RAP, also known as regulator of expression of viral proteins. It encodes for a second protein, P19, which promotes the expression of HIV-1 structural protein by directing unsplice of single-splice viral mRNA to perinuclear clusters where translation takes place. Number 3 is NEF, also known as negative expression factors, which is 25 to 27 kilodalton, was believed to have a down-regulating effect on viral expression. Based on in vitro observations, this view has been revised and several important biological properties have been attributed to NEF. Downregulates MHC1 and CD4 expression, facilitating the invasion from the immune response. Blocks signal transduction pathway by inactivating P56 LCK, a CD4-associated tyrosine kinase, whose activity is essential for activation of P59 FYN, a JAP kinase. Thus, the signaling cascade triggered by T cell receptor occupation in CD4 cell is interrupted. It also promotes viral replications. Now, the HIV and the immune system. The primary cellular targets of HIV are CD4 cells, particularly the helper T cells. Most infected CD4 lymphocytes co-express the CD45RO marker considered as activated or memory helper T lymphocytes. It does appear that activated CD4 T cells are most susceptible to infection. Different HIV strands seem to show different co-receptor specificities. Macrophagitrophic HIV strand, or which predominates in the early stages of infection, use CCR5 as co-receptor. CD4 lymphotropic strand use CXCR4 as co-receptors. Some strains that can infect both CD4 T lymphocytes and macrophages use CCR5 to infect macrophages, and a third chemokine receptor CCR2B to infect lymphocytes. Mucosal transmission involves the infection of both submucosal CD4 T cells and Langerhans cells, followed by rapid spread to the regional lymph node where it propagates to other CD4 cells including helper T cells, macrophages, and dendritic cells. During HIV infection, the expression of co-receptors may be upregulated in cell population that normally do not express them, and that may allow HIV to infect those cell populations. However, some cell populations infected by HIV, for example, intestinal cells, are not known to express CD4. The infection of mucosal epithelial cells is believed to be acquired from infected T cells and probably involves penetration through alternative receptors. Once HIV enters a cell, its RNA is reverse transcribed and the resulting DNA integrates into the host genome, where its expression seems to depend on cellular activations. Stages of HIV infection. First, acute illness associated with seroconversion. The typical incubation time is estimated to be 2 to 4 weeks between exposure and the onset of symptoms. Viremia occurs at that time, and virus transmissions can take place before antibodies are detected. Fever, sore throats, myalgias, headache, malaise, and a mucopapillar rash are the most predominant symptoms. Second, asymptomatic infection. The patient remains seropositive with minimal or no symptoms for variable periods of time, say 9 to 11 years on average, probably 20 years in some individuals. Third, early symptomatic HIV infection, characterized by fever, night sweats, fatigue, chronic diarrhea, and headache in absence of any specific opportunistic disease. In a previously asymptomatic HIV patient, marked the transition towards symptomatic disease. Diarrhea in these patients is most likely due to direct infection of the GI mucosa by the HIV virus. Fourth, late symptomatic and advanced HIV disease. With the progressive decline of CD4 counts, the risk for development of opportunistic infection increases. The onset of opportunistic infection is considered as a clinical hallmark for diagnosis of full-blown AIDS. Transmission. HIV infection is transmitted by 3 main routes. First, sexual contact. Second, significant exposure to infected body fluids or tissues. And from mother to child during pregnancy, delivery, or breastfeeding. There is no risk of acquiring HIV if exposed to feces, nasal secretion, saliva, sputum, sweat, tears, urine, or vomit, unless these are contaminated with bloods. Now, acquired immunodeficiency syndromes or AIDS. AIDS is developed when the HIV infection is left to follow its natural course. The asymptomatic of AIDS may extend for several years. One of the many classifications of the clinical stage of HIV-induced disease is as follows. First, acute retroviral infection. It is usually a mononucleosis-like syndromes. Second, asymptomatic, characterized by no symptoms with the exception of the presence of lymphadenopathy. Number 3, early symptomatic, that is, non-life-threatening infection, chronic or intermittent symptoms. Number 4, late symptomatic, that is, increasingly severe symptoms and life-threatening infections or malignancies. Number 5 is advanced, characterized by high frequency of opportunistic infections or increased death risks. The designation of acquired immunodeficiency syndrome or AIDS is applied when an HIV-positive patient presents one or more of the following features. Number 1, opportunistic infection. Number 2, progressive wasting syndromes, also known as slim disease in Africa, in adults, and failure to thrive in infected infants. Number 3, usually frequent or severe infections not considered as opportunistic, such as recurrent bacterial pneumonia or pulmonary tuberculosis. Number 4, specific neoplastic disease such as Kaposi's sarcoma, non-Hodgkin's lymphoma, and invasive cervical carcinoma. Number 5, neuropsychiatric disease such as encephalopathy or dementia and progressive multifocal leukoencephalopathy or due to reactivation of an infection with Jacobs-Creutzfeldt. Number 6, lymphocytic interstitial pneumonitis in infants and children. Number 7, a CD4 cell count below 200 cubic millimeter. The diagnosis of AIDS need to be supported by evidence of HIV infections, such as positive serological tests, isolation of the virus, or detection of the viral genomic materials by PCR. Serological diagnosis of HIV infection. The initial screening of the anti-HIV antibody is done by an enzyme-linked immunoassay test, also known as EIA, using HIV antigens obtained either from infected cells or by recombinant technology. Since this is a screening test, its cutoff, particularly when used to screen bloods in blood banks, is set for maximal sensitivity, since it is preferable to discard some blood units that test false positive than to transfuse contaminated units with low antibody titers that escape detection. Any positive result on ELISA needs to be confirmed first by repeating the EIA to rule out errors or technical problems. If the repeat test is positive, the result should be confirmed primarily by Western blot or immunoblot. A Western blot is considered positive if antibodies to structural proteins, for example P24, enzyme GP41, and enveloped glycoproteins GP41 or GP120 are simultaneously detected. The accuracy of the combined test, that is EIA and Western blot, is better than 99.5%. Therapy. Starting from the often disappointing experience with single use of reverse transcriptase inhibitors, the additions of new drugs to our armamentarium has proven that antiretroviral therapy can have a beneficial impact on the evolution of HIV infection. First, antiretroviral agents. It may include reverse transcriptase inhibitors such as nucleoside analogues, nucleotide analogues, and non-nucleoside reverse transcriptase inhibitors. Protease inhibitors such as ritonavir, indinavir, etc. Inhibitors of viral penetrations and hydroxyurea. Number 2. Indications for antiretroviral therapy and problems encountered on its implementation. The treatment with multiple drugs combinations, including those needed to treat or prevent opportunistic infections, raise serious problems such as cost, compliance, and drug interactions. Drug interactions become a very significant problem when multiple potent drugs are simultaneously administered. Number 3. Prevention of maternal-fetal transmission. The risk of maternal-fetal transmission of HIV is estimated to be around 25 to 30%, with a direct correlation with the viral load of the infected mothers. The risk can be reduced by a variety of antiretroviral regimens that should contain ZDV, also known as zidovudine. 4th, resistance to antiretrovirals. The high mutation rate of HIV associated with the selective pressure exerted by antiretrovirals results in the rapid emergence of resistant strains. The likelihood of such strains emerging is reduced when several antiretrovirals are used in combination and the patient complies with the prescribed schedules of administration. Number 5. Ancillary therapies. Biological response modifiers have also been widely used in patients with AIDS. Now, conclusion. From the above study, we learned that HIV is a retrovirus which causes AIDS. Immunodeficiency leads to the easy invasion of various bacteria and viruses to the HIV-infected individuals. So, according to the type of bacteria or viruses that invade after HIV infection, the symptoms may be different. We should not panic even if we are infected with the virus. We can take certain measures so that our CD4 may not decrease and live like a healthy man for years. Moreover, we should not treat HIV-infected persons differently. Instead, they should be treated with love and care. Thank you.